Okay, so to start us out today on prokaryotes, I think it would be best that we talk about, again, for starters, the shape and the structure that we would notice in bacteria. And again, this should mostly be review, so we can be fairly speedy through this process. We know that for sure that these prokaryotes, bacteria, would have a cell wall. So I think we should say that there's a cell wall that's present. What is the cell wall made of? Peptidoglycan, right? Peptidoglycan is the primary constituent. It is a protein and sugar complex. Remember that if we have been talking about a eukaryotic cell, what would the cell wall be made of? One of two options, really, depending on what type of creature you are. That's one of them. That's the membrane. Very good. Cell walls made of chitin or cellulose. It kind of depends on what type of creature you have. Cool? Now, with that being said, there is, though informal, there is a mechanism that we can use to classify bacteria based on studying the amounts of peptidoglycan in their cell wall. So that actually, this study actually divides bacteria into two major groups. Bacteria that we would identify as being gram-positive, and then those being gram-negative. So there are the gram-positive bacteria, and then there are the gram-negative bacteria. Bless you. Okay. Remember that if you're gram-positive, or not you, but if a bacteria that you find is gram-positive, then that would represent having more peptidoglycan. Which means that gram-negative bacteria are going to have, for sure, less peptidoglycan. Also, if your peptidoglycan amount is less, that means that your cell wall is not as durable, not as strong, which means that you're more relying, or the bacteria is more, has to rely more so on their membrane to get things or prevent things from coming in and out. So, it's very common that a gram-negative bacteria to have a toxic membrane. And also, I can state that a lot of antibiotics that a doctor may or a doctor can prescribe to an individual, a lot of antibiotics would break down peptidoglycan. So, in which case do you suspect, would it be difficult to kill a bacteria with an antibiotic in the gram-positive or gram-negative category? Gram-negative. So, for sure, this would be a...
you said it breaks up
peptidoglycan, right? So, if there's not much peptidoglycan to break down, that means that like there's other things that you... like this is using exactly. Like in a gram-positive, it's only peptidoglycan. So if you have an antibiotic, there's like only one shield to take down, and you know exactly what it is. But in a gram-negative, there's other like objects that are there for like preventing it being harmed. So in the case of the gram-negative, I would say that antibiotic resistance. What do you mean by that? I'm sorry. In a gram-negative, you mean? Is that what you said? There's like pieces of the membrane that are toxic. And there's certain proteins that are toxic proteins. Mm-hmm. Mm-hmm. Like, what do you mean? I don't know an example. I'm sorry. I really just kind of know the fact. But in all honesty, I don't even want to say this because I don't remember. It's been too long. but I want to say that, like, when you study bacteria, you break them up into 6 or 7 major groups, and some of those 6 groups or 7 groups are positive and some are negative. And like then in each of those cases, like the 3 and 3 or 4 and 3 split up that you have, there are some cases where the bacteria are not harmful to humans, and there's some cases where they are harmful, and there's some of those groups that are a little bit mix and match. But as far as like what an example of who's doing this and how that happens, I'm sorry, I couldn't tell you. Okay, let's talk also about the fact that you might find a capsule on the outside of bacteria. Outside the bacterial cell wall. And this, to me, would be useful in just like overall preventing of disease or issue. Your book has a decent picture of what this looks like. Again, this is all for protection. Some other pieces or other structures that we need to be familiar with. I think one of the most important ones would be the sex pili, which are extensions from the bacteria. And these extensions are responsible for sharing DNA. As you can see, like from my simple little drawing, there's a little sex pili. Information can travel from one to the other. It's not like it could be bi or unidirectional. Now, when they are sharing this information, there's actually a term that talks about how they're sharing their DNA. They're kind of exchanging plasmids. Does anyone know the name of the term that describes how bacteria, if you will, share this information? Do what? Very good, conjugation. Which of course means to share DNA. Bless you.
You okay?
Between bacteria. Now, why would it be valuable for bacteria to share DNA? A resistance. Like let's pretend that I took our bacteria from the other day to Taco Bell. Good for us. And let's say that we did get transformation. So now there's these little green bacteria, and I carry my little petri dish into Taco Bell with a smile on my face. And let's say that I just, I'm all suave about it. I just dump off a little bacteria onto the Taco Bell counter, and I leave. And they don't clean it up. Well, what's going to happen? Well, eventually the bacteria that I leave are going to find the bacteria that are sitting in that burrito. I mean, they're going to meet up eventually. Statistics would say that they're just going to meet. And when they do, they're going to send out each other's pili. They're going to connect, and the ampicillin resistance gene and also the green gene, it's going to get like copied or sent from one bacteria to the other. Now that would leave both bacteria with a copy of this plasmid. Make sense? Sharing information. Like for instance, if Sammy had the inside scoop on how it was going to rain today, and he told me, like he passed just some information to me, right? I was in the unknown. I didn't know it was raining. Just pretend. But would you still know if it was raining? Yeah, like you said, hey, Mr. Fields, it's raining today. Now I know, but you also know. So you keep that knowledge, right? You keep that information. Well, that happens the same way. So the bacteria, they connect up. This guy has something special. This guy doesn't. Okay, now they both do. That's fine. So what are we left with? A bunch of ampicillin-resistant bacteria inside of your Taco Bell burrito, which can only spell disaster, especially seeing as how your doctor might want to, prescribe you ampicillin. Question?
What did you do with the ones that did transform last year?
What did I do?
Yeah, and what did you do with them? How do you dispose of them?
I burned them.
Really?
Autoclave. Safety first. The ones that did transform, right? The ones that didn't, what do we care? Because they don't have ampicillin resistance. But when they do get transformed, autoclave them. So I increase the pressure to like about 8 atmospheres, which is like, if you don't, I mean, if you, it's a lot, okay? It's like being pretty deep in the water. Then I set it to, I don't know, 250 degrees Celsius.
How do you do this?
Autoclave.
Wow.
It's a machine.
Do you have one?
Yes.
Can I play?
Nope. Questions?
Was it fun?
Was it fun?
Yeah.
It's work.
So it could be like pretty dangerous if that bacteria got out.
No, not really. Ampicillin's really not used that much, if at all.
Could you possibly have green burritos?
Yes.
Like on the towel.
But you'd be more likely to get a green burrito just by like visiting a poor Taco Bell than us manipulating the system. Make sense? Pretty good. Like honestly, even though those bacteria are ampicillin resistant, okay, so like they have a resistance for an antibiotic. Let's just say that ampicillin was used like primarily by physicians. Those bacteria that we gained, like they are stripped of any DNA that would cause disease. So those bacteria should be as clean as they could be. They're almost not even bacteria, in that they are not conquering and being parasitic. So they're not your standard E. coli. They're good E. coli. Good for everyone. So yeah. And again, I think the way that conjugation is going to happen when we talk about sharing DNA, I think we want to be specific and state that it's really them sharing their plasmids. Which of course we did the other day. Remember, our plasmid was equal to 2 genes. What were the 2 genes? Hemp resistance and green gene. That's what our plasmid was. The next little piece to mention is motility, which of course means movement. Bacteria are going to move, and as they do that, they'll be using cilia and flagella to facilitate this movement. Usually their movement is fairly limited. Limited in like the fact that they will move. There's not many that do move. And when they do move, they certainly don't move far distances, but then again, they're very, very small. And then we also want to talk about how their movement is usually or typically based on a stimulus. We call that taxis-based movement. They move based on some kind of stimulus that's in the area. Remember that cilia are little extensions off of the cell. The flagella is when you have the major tail. There are the differences between them obvious. They are pretty obvious. Looking at the genetic material of a bacteria, we've already talked about plasmids, which are extranuclear segments of DNA. I've never liked the term nuclear there because we know that bacteria shouldn't have a nucleus anyway, but I think the extranuclear is referring to the fact that it is a piece of DNA that is not native or natural to that bacteria. Remember that bacteria have a single, it just means 1 of course, circular chromosome. And you have 46, so there is this huge difference in number and also just general size of the chromosomes, but then again, we are infinitely more advanced. And this genetic material is maintained, we could say, in the nucleoid region, which is another term that I kind of hate because nucleoid region, like, I would say this classroom is the Mr. Fields region of Pierce High School. Like, it's a non-changing area. Like, these walls, like, encompass my classroom. So region just kind of means that it's an area that's static. But if you think about it, the nucleoid region for a bacteria is really wherever the DNA just happens to be at any given moment, which could be anywhere. So the nucleoid region is like, I don't know, it's not, it's a very dynamic location. It could change over and over. Speaking of their genetic material, when we talk about the process of bacterial cloning, Remember that they use a principle, or they use a mechanism called binary fission. Which again is different than what we use. We use meiosis to generate gamete cells, and then the coming together gamete cells, fertilization causes the new creature. I can do mitosis to clone my body cells, but the cells that I clone are usually almost always differentiated already, right? We talked a little bit about differentiation with Sam Ryan at the genetics concert the other day. Remember that this cloning process can take typically, so on average, it's 1 to 3 hours to get this done. However, some bacteria are quite fast. The fastest time is going to be 8 minutes. But certainly, without a doubt, we could say that they have a very, very short or small generation time. And what will that help facilitate once I say that they have a short generation time? What rate will be exacerbated because they reproduce? Mutation rate, yes, and then what would that lead to possibly? Natural selection, evolution, right? So I would say their evolution is going to be very quick. Cool. Now, every once in a while, you hear of a bacteria that has survived intense times in its little life, and that's because there's also another specialized structure that I need to mention. And this is specialized enough that it's absent in some cases and present in some cases. We want to be careful, make sure we realize that when this is there. The specialized structure that I'm thinking of is called the endospore, which is a structure that is activated in harsh conditions. Which allows the bacteria to become dormant. Like, for instance, if I put them in the autoclave and I thought, man, this is for sure going to get rid of them, and then for some reason we were doing things and the bacteria came in contact with our bodies and they were still, in the event they were still living, then I would say that probably their endospore kept them alive. It's like, imagine that it was like third world country situation, and that we had 2012 happened, or whatever, and people have their little bunkers or whatever, and they take their can of pork and beans from 1965, that big can of your grandpa bought. That would be like our endospore. We're just kind of waiting around for things to get better, or with the hope that things might get better. So it's like going to sleep and waiting till things are just right. And some bacteria can do that. Cool?
Yeah.
Okay.